This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 88, recorded on June 25th, 2010. Hi everybody, it's Vincent Racaniello, and this is TWiV. Are you ready for some virology? Well, doing it with me today from Western Massachusetts is Alan Dove.
Good to be back.
How you been, Alan?
Oh, doing okay.
You growing vegetables up there?
Well, I tried a little bit, but then, the varmints got them.
Oh, rabbits?
We've got, we have rabbits, we have chipmunks, across the street I've seen some deer, so out here.
The deer will eat everything.
Yes, so we're out here in the wilderness.
Can you see a house nearby you?
Oh, yeah, my neighbors are all visible. It's a suburban development, but, a lot of trees around and plenty of critters.
Also joining us today from a little farther west in Cleveland, Ohio, Marc Pelletier. Hey, Marc.
How's it going?
Welcome back to TWiV.
I'm just catching up on the last paper that I had to read for this. I feel like I'm just cramming for an exam.
It's back to graduate school.
Okay, it's done.
Well, you don't have to read it. You could just ask questions,
Sure. No, these are amazing papers, though.
They're great. Marc, of course, is from Futures in Biotech, twit.tv/FIB, but his real job is Chief Scientific Officer of Aeromics, LLC.
Yeah, small little biotech company working on drugs to treat brain swelling.
That's what pays the bills, right?
and all this podcasting gear, too.
Well, that's okay. I listened to your last podcast on the heart stuff, curing heart problems with viruses or drugs instead of surgery.
It's pretty hardcore. We had a young assistant professor who's across the street at Case Western, and he's really, really, really smart. And he very well. So one of the issues with that show is that I've had heart surgery, and I've kind of tap danced around, never having not done an episode on anything heart biotech. And after 60 episodes, it was time to actually tackle the hard stuff. And so I brought him on, and he explained two really cool examples of some of the biotechnology that's going on right now to treat heart disease, which is the number one killer. And humanity's number one killer. So we're talking 1 out of 5 people. Yeah. And there's some viruses involved. So that was really, really cool.
I had no idea that kind of thing was going on. So I really enjoyed it.
Yeah, gene therapy.
And one of our, of course, we have a story on gene therapy today for a different disease. And we also have a story inspired by your episode, which we'll get to a bit later. But it was that book, Darwin's Radio, that you did an Audible ad for.
It's awfully creepy.
And I found it. I thought, we have to do this story. He'll love this. So we'll do that. That'll be story number 3. So we have 3 stories today, and let's get right into them. The first one is a fun story, and it came across this just a few days ago. It's controlling a rhino beetle with a virus. This was a story I found in the Pacific News Center. And this takes place in Guam, where they have many coconut palm trees. And apparently there is a beetle, a rhino beetle, that is decimating these trees. So they've tried insecticides to get rid of these. These are these big guys, 1 or 2 inches long with a big horn coming out of the front. I saw one just the other day in my backyard, actually. So we have them here in New Jersey as well.
There's a movie called Bug's Life by Pixar.
Yes.
He plays one of the characters.
Yeah, that's right. Was he a good guy?
He was a good guy. Very roly-poly, likable character.
What was his name in the movie? Do you remember?
Oh, I don't know. I've only seen it 10 times. Right?
It could be an episode title. That's why I'm asking.
Oh, no, I don't know.
Anyway, rhino beetles, they go up into the crown of the palms and they wreck them. Now, it's not just an aesthetic problem, because apparently there are companies that make palm oil from these trees. I didn't know that. I don't know what you do with palm oil. I guess you eat it, right?
Flavor things.
Put it on salads.
Yeah, I think it's used in a lot of products.
So these beetles are wrecking this industry. So as I said, they tried insecticides, but what they're doing now, and seems to work, is to use a virus to control them. And the article, I don't know if you guys are looking at this article, but this is what drew me to this story. Dr. Aubrey Moore, who is a scientist on Guam at the University of Guam, he's sitting at a table. He has a couple of rhino beetles on the table in front of him. And he's got bare hands. He's got a pipetter in one hand and a vial of what probably is virus. And he's pipetting the virus into these beetles. And he's wearing this great Hawaiian shirt. He just looks like he's having a great time.
These things are the size of golf balls, too.
They're huge rhinos, and he's pipetting. So apparently what they do is they infect them. They put the virus in their mouths or mouth parts. And they wait until the virus grows in them. And then they release the rhino beetles out into the wild, and they spread the virus to other rhino beetles. And apparently it keeps their numbers down. It doesn't eradicate the rhino beetles, but helps control the disease.
Well, I guess they're trying to control the top of the insect food chain here.
Yeah.
There's... In traditional, biological control of pests, you might see them bring in ladybugs or other small insects to eat things that are even smaller. But what do you do when it comes to the biggest bug?
The big ones. No, this is an interesting approach. I mean, I don't know of using viruses in this way very much. Usually it's an issue to release viruses like this. So I'm actually a little surprised that this is going on. They've tested this in Malaysia before, which I think is where the beetle originated and where the virus is as well. So this is a naturally occurring virus of rhino beetles, and they have taken it and used it to their advantage. So apparently when you infect the beetles with the virus, it damages their stomach walls and they stop eating, which is...
Should we be concerned about it spreading to other insects?
That's a good question. I mean, I found a few papers that insist that it's very species specific, but I don't know. I don't see why it couldn't.
But we haven't
change.
identified all the species of insects, particularly beetles.
Yeah, exactly. And I'm sure they haven't tested many either, right?
Right. I think they're the most diverse order, if I'm not mistaken, right?
Yes. What is it, that book?
An Inordinate Fondness for Beetles.
Exactly, yes.
How quick is it to identify the receptor of a virus?
Can take years. Can take decades for some viruses, and we still don't know.
Because if you could identify what the receptor is, and you could say, well, yeah, this virus is specific for this beetle. Yeah. There's no homologue of that gene for the protein that's the receptor in any other insects that we've seen. And you can go into the community of insects, crush them all up, right, as a big ball of goo, and then do and sequence the whole thing and see if the receptors are there.
I don't know in this case.
Right, but that still doesn't tell you if the virus can use other receptors.
Oh.
Yeah.
So because in a pinch, they will adapt.
Yeah, they could adapt, or they could use multiple receptors, sure. So
Aubrey Moore is okay, so it doesn't affect humans.
Doesn't seem to. I would guess it would not. So this actually brings us to the virus itself. This is a member of a rather small group of viruses called nudiviruses, N-U-D-I, for naked. I don't know why they're called naked, because they're actually enveloped viruses, but so be it. And they are comprised largely of viruses of invertebrates. There's the rhinoceros beetle virus, the palm rhinoceros beetle virus, which is the one they're using here. There's also a cricket virus and a corn earworm moth virus and a shrimp virus, all invertebrates. And these are large DNA-containing viruses.
If it doesn't spread beyond, if it doesn't eradicate the population of beetles, then maybe it's probably fairly safe in that it's not that infectious a virus.
It could be.
Well, I guess it could be to another insect.
Yeah, I don't see why it might not, and I doubt that they're looking. I don't know if Dr.
Right, but the other thing is, if this is a naturally occurring virus in the rhinoceros beetles in their native land in Malaysia, then presumably other insects have already been exposed to it, and the ecosystem has already reached its conclusions about this virus.
Makes sense.
It makes sense, but then
But then you've introduced the beetle into some other place
Yeah.
like Guam, and what else there has seen it? Yeah,
Right.
it does
Exactly.
raise some issues. I'd be more concerned that the effect of the control regimen that they're implementing here would wear off. I mean, something
Yes.
like this has been tried with rabbits in Australia. We talked about that just recently, right?
Yeah. The Myxoma virus was introduced into Australia to get rid
Yeah.
of rabbits, and after a year, they all became resistant, and the virus became less virulent. So
Right.
that's one of the things I wanted to bring up here, that could happen easily. In fact, most of the papers I've seen say that the use of the virus never eradicates the beetles. So it could be that some propagate that are resistant. So there are a number of issues, but maybe it keeps it low enough so that it's not an issue, and then you can combine that with other approaches and, keep your palm industry intact.
Let me ask you a question.
Sure.
Is Aubrey Moore the only person who studies gut viruses of rhino beetles?
No, there's a few others. I mean, it's not a huge study, but if you go to PubMed, you get about a dozen papers on this virus with various groups. I'm looking at one here that is called The Incidence and Use of the Virus for Control of Rhinoceros Beetle in Palm Plantations in Malaysia. And this is from, some of the authors are from the Malaysian Palm Oil Board. So this is where the virus was first identified, actually, in Malaysia, and used there, and then in a few other countries, Guam and a variety of other southern Pacific and Indian Ocean countries.
How do they identify it? Do they find a bunch of sick rhino?
I would guess
beetles
that's it. Yeah, they probably found some sick-looking ones and identified the virus. That's typically what happens, unless you go looking specifically in insects. You could do that as well, but often that's for a virus that I know of, cricket paralysis virus. They found paralyzed crickets in Australia and found that they were infected with a virus. So, this virus could change. The beetles could change. Anyway, I thought it was an interesting example of one use of viruses to control an agricultural pest. I think it's pretty interesting.
So it's a plus. You endorse it or not endorse it?
I think if you keep it down there where the palms are infested, and if you monitor what's going on, I think it's probably fine. I don't think it's a threat for people. What do you think, Alan?
I think it's probably fine. As I say, it's a virus native. If it's a virus native to the native land of this beetle, and the beetle's already there, ideally you wouldn't have introduced the beetle in the first place into an alien environment. But now that you have it there, you might as well bring the parasites that prey on it. And hopefully they did test as many local insect species as they could find and see if this thing could infect them. But at a certain point, you have to weigh that against the amount of destruction the insect is doing.
Yeah.
You just have to test the beetle.
And also weigh it against the real world options. I mean, people can say idealistically we should never introduce something like this, but out here in the real world, what's going to happen if you don't reduce these with a virus is farmers are going to go around and spray noxious chemicals to get rid of them, and which is worse.
Yeah, that's one of the issues that's mentioned here, that the chemicals used to control these beetles have worse issues than possibly the virus itself. So
if using a ladybug is considered organic form of pest control, is using a virus organic pest control?
Well, using BT toxin is considered organic.
Yeah.
So I would think this would be okay.
I mean, when I used to have a lawn, I used to put BT spores on the lawn to kill, I don't know, fungi or something. I don't remember what. And that was perfectly fine. So it's a biological control mechanism. People, I think, have issues with viruses because they
make good movies.
they have a mind of their own. They do what they want.
And by the way, the rhinoceros beetle in A Bug's Life was named Dim.
DIM.
DIM. It's voiced by Brad Garrett.
Good.
Thank you, Wikipedia.
Yes, good job. Maybe you can work that into an episode title, Alan. So these are related to baculoviruses. You might have heard of baculoviruses, Marc.
I've made a bunch of them.
Yeah, we use them to express things, proteins. They're very good, and those are
complexes too. You can mix a couple of viruses together.
Yeah.
Form protein complexes with them.
Nice insect viruses. Baculosis.
Spodoptera.
Spodoptera frugiperda. is one of them, yeah. So these are related to those viruses. They have big circular DNA genomes. They are 100 to 200 kilobases long, a few hundred proteins. Not huge, but big. Interesting viruses. Okay.
Aren't there baculoviruses that infect human cells? I'm sorry about the
I think you
sidetrack
can.
there.
I think you can infect human cells with baculoviruses. Yes. For many years, we used to use insect virus cultures to express proteins in baculovirus vectors, but recently it was found that you can do it in human cells. That's right, mammalian cells.
With the insect virus.
Yep, with the insect virus. So it recognizes a receptor in mammalian cells.
And this is a baculovirus.
That's related to a baculo. So yes, your point is well taken. They could infect.
It's a naturally occurring one, though.
It's an easy enough experiment to do. Aubrey Moore here could do his work in a hood rather than out on a table and see if they infect mammalian cells.
I don't know if it has a hood there. It's got a bucket. You see a picture of a bucket in the back.
Yeah, this is a great picture. We're going to put a link to this because this is just wonderful. It's just classic. I don't think he really cares. He seems like he wants to go out and do a barbecue pretty soon.
Well, they're meaty bugs too.
They're huge.
Yeah, I was kind of wondering, can you eat these?
Toast them.
I'm sure somewhere, if you're starving, you're going to eat anything, right?
Well, but some insects actually don't taste too bad.
Yeah, there's probably a lot of protein in it, right?
Yeah. Yeah, and the genus name for crickets is Grillus.
Mm-hmm. Grillus.
So they have instructions written right on them.
Excellent. The funny thing is, I'm looking at this story in the Pacific News Center, and there's an ad for margaritas, burritos, and barbecue on the right. This actually, just parenthetically, I don't know if either of you have seen this in the past week or so. There have been some, probably you don't because you don't search the news for virus, but a cricket farm in Florida was wiped out by a virus. So this is a cricket farm that's used to supply pet houses because many pets eat crickets. So you go and you buy crickets every week. If you have, for example, what are those lizards that with the long tongues that catch insects?
Chameleon.
Chameleon. You feed them crickets. You go to the pet store and you buy them. And this was apparently one of the major suppliers in the U.S. and they got wiped out. Like 9 million crickets killed by some. I think it was a parvovirus, and they're out of business.
Wow.
Very interesting. So these viruses not only infect people, they infect everything. Everything that is alive has a virus that will infect it. Okay, that is controlling the rhino beetle with a virus. Now, the next one is a bit more serious. And this is a recent story that came out in Science Translational Medicine, an RNA-based gene therapy for AIDS. And this is a very interesting story that actually builds on a number of stories that we've talked about on TWiV over the past year. So let's see. Everyone knows that AIDS is caused by HIV-1. We can treat AIDS with HAART, highly active antiretroviral therapy, which is a combination of 3 antiviral drugs. But it never cures the infection. You're always having virus in you. You have to take these drugs all the time, every day. And so it's not the ideal therapy, so people are always looking for something else. So that's the basis of this paper.
If somebody wants a review on HIV, I did a show, Futures in Biotech episode 32 with Ronald Coleman.
Yeah, good one.
And they can go back, and he describes HIV down to the to its infection, the whole life cycle, everything about it.
That's a good one. I remember that.
I think that's how we met, though, didn't we? You had listened to that one and sent me an email?
I think the way we met was I named you a pick of the week. I named Futures in Biotech an early pick of the week on TWiV.
Cool.
And you were one of the few picks that ever responded and said thank you. So we started a conversation. So one of the things that... So HIV infects, among other cells, CD4 positive lymphocytes. So the idea has been arising that maybe we can somehow modify lymphocytes in people and make them immune to AIDS or unable to be infected with HIV. And you probably both remember that not too long ago there was a patient, I believe in Germany, who had AIDS. He also had leukemia. And so he was given a bone marrow transplant. And the donor that was used had a mutation in the gene coding for one of the co-receptors for the virus.
CCR5.
Yes. So this protein is needed for virus infection. It goes along with CD4 on the cell surface, and it's required for virus to get into cells. But there are a number of people, a few percent of the world's population have a mutation in this gene, so they don't make the protein, and they're actually resistant to infection with the virus. So this fellow in Germany was given a bone marrow transplant from someone who had a deletion in CCR5 gene. And he no longer has HIV in him. He has no more virus. And it's been, I don't know, a year or two now. So that gave everyone the idea, maybe we could give people modified, not T cells, but precursors to T cells, which are hematopoietic cell precursors. So you can take cells from the blood and from people, which are the precursors of all the blood cells, of all the lymphocytes and macrophages, etc. If you could modify them, put them back into the people, maybe they would be resistant to HIV. Okay, so that's the idea. And the proof of concept is the German patient getting the CCR5 deleted bone marrow transplant.
Sure. And there, by the way, I think the development of CCR5 antagonists for the interaction, right, for the viral.
Yeah, there is actually a licensed drug. It's called Mariviroc. It is a CCR5 antagonist. It's a small molecule that binds the protein and deforms it and prevents it from functioning as a co-receptor. So that is one of the components of some heart combinations. Yeah.
But that might not be as effective, right? Because you got to get the dose. There might be side effects.
Yeah, it's one of a couple of drugs you have to take, and it's expensive. So if you could make people genetically resistant, it could be better. So I mean, I don't know how far off this is, but as we see in this paper, people are working on it. So what they did here is a very interesting experiment. They took AIDS patients who had lymphomas. So it's AIDS-associated lymphoma. At some point in the course of AIDS, a number of patients do develop lymphomas, that is, cancers of the lymphocytes. And in this case, what they do for these patients is they take out from their blood, they purify the hematopoietic precursor cells. And then they ablate the patient's bone marrow, basically, and then they infuse back into them the cells that were taken out, the precursors. So the idea is that the precursors are not infected and they're clean, so to speak. You destroy all the existing lymphocytes, which are the sites of HIV replication and all the lymphoma, and then you give them back their own hematopoietic precursor cells. And they repopulate the patients, and now they have no more lymphoma.
And no more lymphoma, and hopefully no more CCR5. It says what are the genes that they express? 3 RNA-based anti-HIV moieties.
Right. So what they did is, when these people, they took out their hematopoietic precursor cells from the blood, and they split them in half. In half they didn't treat. In the other half, they infected with a lentivirus that expresses 3 different anti-HIV RNAs, basically. So these are targeting CCR5. Basically, it's a ribozyme, which is an RNA enzyme that will cleave the CCR5 mRNA. They have a short interfering RNA, which targets a viral mRNA that encodes an essential protein called TWiV. Mm-hmm. And then they have a third RNA, which is a RNA that binds a viral protein that's required for transcription. And it basically sequesters that protein and prevents it from functioning. It's called a TAR decoy.
That's really cool. I'm looking at the CCR5 ribozyme. So they're not trying to knock out the CCR5 from the cell, but they throw in an enzyme that will kill the RNA made for CCR5. That's wild.
This approach, this triple inhibition, and it's using a lentivirus to infect T cells, that's been shown before in culture to totally prevent HIV infection. So you have 3 different approaches, and using 3 is good because the virus will always mutate to avoid just one. And if you have three, it's a lot harder for the virus to get around this. Just like why we use three drugs, we use three antiviral RNAs. So they take the hematopoietic precursor cells from these patients, infect them with the lentivirus that has these three anti-HIV RNAs. They grow the cells a bit in culture, and then they put them back in the patients. Now, they say here is very interesting. Ethical considerations prevent them from just giving them back the modified hematopoietic precursor cells.
Why is that?
They have to give them both. Well, they know that the untreated precursor cells will cure them of their lymphoma. So you can't hold that back from them.
Right.
And we don't know if the genetically modified precursor cells will adequately repopulate those patients. So you could be giving them a death sentence, basically, if you only gave them the modified one. So they have to give them a mixture to make sure that they get cured at least of their lymphoma. The downside is, of course, that they're going to have at best a mixture of HIV-sensitive and HIV-resistant lymphocytes in the end, right?
Mm-hmm.
Right.
So it's not perfect, but it's just an exploratory study. And since they're already giving these patients a transplant of their own precursor cells, they figured, let's try this and see what happens. So it was approved. The patients have to agree to do it. And it turns out that these cells will persist. The cells containing the anti-HIV RNAs do persist in these patients for a long period of time. I think they looked out for 2 years. And at least in a couple of cells, they have a small fraction of lymphocytes. Again, these lymphocytes derived from the precursor cells that have the anti-HIV RNAs in them. So it's a proof of concept, basically. Now, we know nothing about HIV because all these patients are on triple therapy, and they don't have any detectable virus in their blood. And you can't take them off triple therapy and ask what happens with these, inhibitory lymphocytes, because that's not ethical either to do. But at least you know that these modified cells will make lymphocytes in these patients. Remember, you're putting in a precursor. You're putting in a hematopoietic precursor cell, and you're depending on that differentiating into all the different blood cells, lymphocytes, monocytes, neutrophils, and keeping these antiviral RNAs in them. And so, remember, the CD4 T lymphocytes are the ones that are going to be infected with HIV. So the assumption is if these antiviral RNAs are there, they would be resistant to HIV.
Now, why did they use lentivirus to introduce it to these cells? Is it just because they infect hematopoietic progenitor cells pretty well, or?
The lentiviral vectors are the ones that have been the best developed now. They will infect a wide variety of cells. It doesn't matter if they're non-mitotic or not. They don't have to be dividing. So they will infect a wide variety of cells. So they're being used for a lot of gene therapy experiments. They're approved for a variety of applications, so it makes sense to use that for this.
And they introduce their DNA, or they introduce the gene stably into the chromosomes.
Well, yeah, they integrate. These are retroviral vectors, right? They can't make particles, but the DNA will integrate, right? So,
so it's stable.
It's stable, and so within 2-year frame in this experiment, some of the people are still expressing these antiviral RNAs.
Now, they're not in germ cells, right? So
they're not in germ cells, no.
They won't pass down through 2 million years of human.
No, no, they won't. That would be interesting, wouldn't it, if you could do that, and then all the offspring would be resistant to HIV. Someday, perhaps we'll do that.
But one thing that is interesting here is since these cells are persistent, it may, if you have enough patients who you have these cells persistently, the modified cells, then in a future study, you may be able to lift the ethical requirement of giving them the untreated cells as well.
Yeah, sure. If you can show that the cells that...
if these cells are capable of repopulating them and treating the lymphoma,
right,
and you have pretty good evidence that that's the case, then you might be able to do the actual experiment of just giving these cells. But it would take a few more years of monitoring because, as you said, otherwise that would be,
Yeah. No, you're
I
right.
tested in monkeys. That's a simian experiment. Let's get out the monkeys and do that first. I suppose.
Sure.
That'd be great.
Yeah, so you
You
have
take
to
a-
show that these precursor cells that have been infected with your lentiviral vectors are functionally competent to do everything that they need to do. They make all the different cell types in the blood, and they're functional. And that
Right.
is,
Well-
it's going to take a while to get all that data, right?
While they're at it, say the patient's also diabetic. Could you throw in insulin? I mean, would these hematopoietic progenitor cells be able to secrete? I don't know. Why 3 genes? Why not fix all their genetic defects, right? If
I don't know.
Well,
if they're-
there are some things that you could fix in lymphocytes and some that you couldn't.
Sure,
Yeah.
we could use lymphocytes as a
Yeah.
source of insulin. I don't know. I'm just
It's no.
Okay.
It's certainly If somebody is insulin sensitive and diabetic, then that might be an approach.
Yeah.
Yeah.
So two more interesting things here that I learned. One is that, the original German patient, that was a transplant from another patient, and that's not practical because you have to have a match between the donor and the recipient. So not only do you have to have a CCR5 deleted donor, but they have to be matched with the recipient. So that's apparently very hard to do. Here, you're getting your own precursor cells back, so it's a lot easier to do. And these precursors are purified from the blood. This is actually a relatively recent development, so you don't have to take the hematopoietic precursors out of the bone marrow anymore, which was a difficult procedure. And now you take some blood. You take actually multiple blood bleeds over the course of time, and you purify the precursors from the blood. You propagate them in the presence of certain cytokines, and then you can give them back. So it's been made possible by a lot of other developments in medicine that have nothing to do with viruses.
you could be allergic to the CCR5 ribozymes, but that you just test it, right?
Yeah, that's a good point. So they wanted to know if any of these RNAs were toxic in any way, and so they did some limited blood work on these patients, and they said there didn't seem to be any problems in any of the blood cells that are produced from the progenitors. So it looks
Well,
okay.
it might also be interesting. Now, these progenitors are also going to give rise to CD4 positive cells, right?
Sure. Yeah.
So you're going to have two types of CD4 cells floating around, some infectable by HIV and some not.
Right.
So if these patients do, obviously they're all under heart and they're not going to have detectable virus, but if the virus does get back and start infecting CD4 cells and their CD4 counts go down, you would expect them to selectively depress the unmodified CD4 counts.
Exactly. That's a very good point. In fact, one of the patients in this study went off his heart. He stopped taking his drugs.
He left it in San Francisco.
He left it in San Francisco. So he had a rise in detectable virus in the blood for a short period of time. And they said in that patient, the fraction of CD4 cells that were containing the anti-HIV RNAs went up. Not a lot, but it did go up. So they said it's only one patient, so we can't conclude anything, but it could be that exactly as you're saying, you get rid of the susceptible cells and you enrich for the resistant ones. So yes, I don't know how you would use that because you can't take them off heart, but
if any of them happen to go off heart though for a longer period, they could end up doing a very interesting experiment. I'm not advocating that. If anybody in that study is listening to this, don't go off your meds. But if somebody did that and it wiped out their unmodified CD4 cells, and then their modified CD4 cells came back, where would the virus go?
Yeah, exactly. And in fact, I understand that many patients on heart stop taking drugs.
Well, the drugs are nasty.
They're nasty. You have to take them every day. It's costly.
The schedule is horrendous, and it's expensive, and it's I'm certainly not. I don't condone going off medications like that, but I can certainly understand how somebody might.
Right.
Well, you never know the circumstance of the patient where there's a reaction or cross-reactivity. You might need another kind of pharmacological treatment for some other problem, and there's an indication or that there's a cross-reactivity between the drugs. So he has to pick which one he has to take. Well, this one might be okay to drop. It might be the safer one to
right.
It's up to the doctors to decide.
These are very sick patients to begin with.
Yeah.
Seems like I'd want this. had to pick, or my kids, if they had HIV and they could potentially have some transgenic hematopoietic progenitor cells, it'd be an extra line of defense, which would be amazing.
Yeah.
So keep an eye on this. I think this is going to go forward and be developed into something useful. I mean, this kind of transplant isn't trivial, and there are a lot of side effects. There are infections and potential things that can go wrong, but if it turns out to cure AIDS, then it could be widespread.
Yeah.
This was at the City of Hope, Duarte, California.
Let's see. I just closed the paper. You'll have to tell me. Yes, I think so. I think that was one of the institutions.
David L. D'Augusto.
Yep.
First author and last author is John A. Zaya. So Z-A-I-A. John A. Zaya.
All right, this third story, Marc, is for you.
Cool.
This is because you talked about Darwin's Radio. And if I remember, I haven't read Darwin's Radio. Did you read that book, Alan?
No, I didn't.
It is about a virus a virus from the germline of humans that somehow comes out and starts infecting people, right, Marc?
Oh, they bring it back.
Someone
they
brings
do
it back.
exactly what this paper says.
So in this paper, they actually did it. So I thought it would be really cool to talk about this. This is a paper from Paul Benash's lab here in New York City. Young Nam Lee is the first author, and it was published in 2007 in PLOS Pathogens. The title is Reconstitution of an Infectious Human Endogenous Retrovirus. So as we know, when retroviruses infect cells, they are RNA viruses. The RNA is converted to DNA by a viral enzyme, and the DNA integrates into the chromosome of the host, and it's there permanently. As long as the cell is living, that retroviral DNA is there. As a consequence, humans and many, many other animals have in their genomes records of their infection. So if these viruses infect germ cells, sperm or oocytes, the DNA is passed on to subsequent generations. So there are such endogenous retroviruses, as we call them, integrated in our genomes and those of many other animals. And in this paper, they say endogenous retroviruses comprise about 8% of human DNA. So all of these are DNAs that have been with us for millions of years, and not just hominids, but our precursors, our evolutionary precursors.
Let me ask you. So 8%, if you think about it, when a chimp is 99% human, right? Or we're 99% chimp. So
yes.
So that 8% means that DNA is really ancestral to before speciation, human speciation.
Well,
Yes.
some of it is.
Some of it, yeah.
I guess a lot of our DNA is from before speciation.
Sure.
Think
Yeah.
about it.
Yeah.
Okay. So it's not
No, it's true. I think the difference between humans and chimp is all retroviral. That's what I've heard said. But anyway, so there are lots of endogenous retroviruses in our DNA, but they're all defective. That is, they have mutations, so they don't make viruses. None of us are producing. None of us are producing infectious retroviruses. But you can sequence them. When the genome was sequenced, you could see all these, and they look like retroviral genomes, and you can see all the mutations, and in theory, you could fix them.
Well,
right?
so let's do it.
So let's do it.
Guys.
So that's what they did in this paper. They took one called HERV-K. So HERV is human endogenous retrovirus, and K is just a certain kind of HERV. There are many of them. Okay. And these are interesting because these seem to be very recently deposited in our genome. And they may have been replicating 1 million years ago, are the estimates. So one thing I thought was interesting in this paper is how you can use the retroviral sequence to determine how old the endogenous retrovirus is. So what happens is when these viruses integrate into your DNA, the viral genome is duplicated at either end as what's called an LTR, a long terminal repeat. So it's the same sequence at the left and the right end of the genome. And then that stays in the chromosomal DNA. And then you can compare the two sequences over time. They should be mutating independently, right? And they say here that there is one difference between two LTRs should occur every 200,000 to 450,000 years. So by comparing the number of differences in the two LTRs, you could figure out how old they are in the genome, which I thought is cool. So this one is about maybe 1 million years ago, or slightly less. These are HERV-Ks. So what they did in this paper is they took HERV-K sequences from 10 different individuals and said, where are all the mistakes, and let's correct them. And they made a virus that is correct. Okay?
And as Pandora said, oh, I'll just open it. What could go wrong?
Yeah. Poke, 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 poke.
And this is here in New York City, of course.
By the way, I hate to plug the Futures in Biotech all the time, but I guess that's kind of why
That's okay. You can do it.
So Futures in Biotech episode 34, a great historical document, the human genome. I have Marc on from Yale, who's a phenomenal bioinformatician, and he goes into the human genome's human genome in terms of historical documents. But anyhow.
So that's 34? Cool. That's one I haven't listened to.
Gerstein's fantastic.
All right, I'll do that.
He probably listens to this show. So
I don't think so. Probably not.
I think so. He's a huge podcast listener.
So these guys reassembled the whole genome, basically. They put it in a plasmid, and they put it into cells, and the cells make virus particles. So now
How much did they have to fix?
So there were 10, let's see, can I find that number quickly? Not too many. If you look at figure 1, I would say they're less than 50 for sure.
Just subtle changes.
Yeah, single point changes.
How did they know what was conserved again?
So they took 10 different sequences from 10 different individuals.
Right.
And they say, here's what they say actually. It's all these encoded an obvious defect in at least one of the viral proteins. So in other words, a base that would shift the reading frame or terminate the protein, and they just fixed it. So every one of these 10 had a defect, and in at least one. So they took the good ones and then fixed the bad ones, and then made one virus with correct sequence.
Right. So they essentially constructed a phylogenetic tree, traced it back to when there was probably a functional virus, and built that virus.
Yes, right. Exactly. And they say that the virus they ended up making is at the root of the phylogenetic tree of all these 10 endogenous retroviruses. Yeah. And this makes viruses. You could see them. They have a picture of them in this paper. The only caveat here, all right, so retroviruses are enveloped. The core is composed of structural proteins, and then the membrane has in it glycoprotein called envelope glycoprotein. These viruses didn't work with the envelope glycoprotein from the HERV viral genome. They were not infectious. So these are not exactly HERV-K viruses that they made. They had to put another viral glycoprotein in there to get it to work, and they used the glycoprotein from vesicular stomatitis virus, VSV G protein.
Mm-hmm.
So it's almost a HERV-K virus, but not quite, because it doesn't have the HERV-K glycoprotein. And maybe that's good. Maybe it would be problematic if it had a HERV-K glycoprotein. So this is a virus that hasn't been around maybe for 1 million years or a few 100,000 years, and it's in a lab, and you can study it. So that's pretty cool. But anyway, Marc, maybe that's where Greg Bear got his idea from.
Maybe that's where these guys got their idea from.
Could be from Greg Bear, yeah.
So, but I guess one advantage is that if ever we see this virus again, we have the tools and heads up as to how some of the molecular components and strategies to block it.
Could make a vaccine, right? Yeah.
You could, maybe, now that you have a heads up. But the other side of the coin is, again, poke, 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 poke.
Right, and we've been kind of joking around flippantly about this, but seriously, since this is a virus that infected our ancestors, or something very similar to it infected our ancestors a couple million years ago, this is something that our collective immune systems have already made peace with.
Exactly.
So it's not
years ago, though?
I would think if it were going to wipe us out, it's had a chance to do that.
It had a chance, exactly. I think that's a great point, because we have this endogenously in our genomes, in our germ cells, and so obviously it was infecting us at one time, and we're fine. So, in fact, human genomes encode a number of proteins.
I mean, we say we're fine. Maybe we've never been the same since.
Yeah, maybe this is the seeds of our destruction.
That's right.
This is the apple, Adam's apple. So,
but also, this is not the first reactivated retrovirus. This may be the first one in humans.
Yeah.
There's Sleeping Beauty is the one. I finally remembered what it was with the aid of Google. There's a zebrafish endogenous retrovirus that was reactive, or transposon, I guess it was, that was reactivated this way.
That's right. Yeah. No, and there's another interesting aspect of this is that human cells make a number of antiretroviral proteins like ApoBec and the TRIM proteins, and it's believed that there has been some positive selection for those proteins to be retained over evolutionary time, and it could be that viruses like this at one time provided the selection for that. So those protected us against HERV-Ks or something related to it. So it's very cool. I mean, this is really neat stuff. This is something really old, and now we can study it in some form.
Yeah.
I think it's terrific. So there you go, Marc. That's reality.
Well, you've done challenging experiments. Even your first big experiment, right, which was bringing back polio. Or not bringing back polio, sorry, engineering it.
We engineered polio, but we never... Yeah, neither this experiment nor mine actually made a new virus. I mean, this HERV-K, the genome is there. It was just a little screwy, and all they did was correct it. But no one has made a totally new virus, by my definition, which would be one you've never seen before. I don't think you could do that.
What about the chances that this happens spontaneously from our genome? Could an ancient virus? I mean, I'm looking at the number of mutations here on Figure 1.
Oh, could it spontaneously be corrected? That's a good question. I was thinking about
that. With 6 billion people out there, 1, 2, 3, 4, 5, 6, 7, 8, 12.
I don't know. It would so that would have to be random, right? Because there's no no selection for it.
Right.
But that's another argument in favor of the safety of this thing too.
Right.
Yeah.
Is that these mutations are going to flip back and forth at some rate. And over time, enough monkeys, enough typewriters, it could reactivate on its own, but it hasn't, or it hasn't done any damage by doing so, right?
Well, these guys have already brought it back, so
and now these guys brought it back, but I'm not terribly concerned about this escaping the lab. All kidding aside.
Yeah.
Right. No.
Now, on the other hand, in Greg Bear's book Darwin's Radio, this escaped virus does do harm.
Well, viruses have to escape if it's going to be good fiction.
That's right, and it is fiction. So I thought you
really interesting paper. Yeah, it's fascinating. The idea to go back into our history and bring it back to life. I mean, what other genes could you bring back to life that are ancestral genes? Right. We did an episode a few episodes back with a person who, what was it called? An episode where we what's it called? 10,000K, Mapping Life's Greatest Journey. So Project Genome 10K, the idea is to sequence every mammalian genome, and or 10,000 mammalian genomes, I think it is. It's David Hostler. So this guy, he was the first to assemble the human genome. So when the genome was sequenced, he had all the data and he assembled it into the map that it is. And he held the disk. He's the first person to hold the human genome in his hands. So he was saying that by looking back, through all our genomes, all the mammalian genomes, comparing them, what is common to all of us is ancestral, right? So it's looking back into our genetic history, and maybe there's some stuff in there that's dormant, right, that we could bring back.
Yeah, I don't know of any examples of genes that are incomplete or mutated. Do you know, Alan, offhand?
Nothing springs to mind.
Yeah, there might be something, but you could, in theory, if you have some way to fill in the broken parts. Yeah, you could do it.
We'd have to bring Gerstein back and have him go over, because he's been doing it for it's 3 years. It's our last episode, 2.5 years.
Yeah.
maybe he could give us some light as to, because I mean that's what they're doing. They're looking for genes that are quiet, dormant, and
Sure.
be interesting. So these guys brought some dormant material back from millions of years ago.
Kind of sounds spooky, doesn't it? Bringing back the dead. Of course, in terms of a virus, they're already dead, so.
Are they? Oh,
not dead, not living.
I'm going to get a lot of argument. I know, I'm just teasing.
That's good.
I'm just teasing. All right, that does it for our stories. Let's do a couple of emails, but before we do, I want to remind everyone that this episode of TWiV is sponsored by Data Robotics, the makers of Drobo, wonderful backup and storage and working solutions for your computer. Drobos are, well, they look like toasters, as you'll hear in an email in a moment. They're wonderful little boxes that can hold 4, 5, or 8 hard drives, and you simply plug them into your computer, copy your data to them, and the good thing is your data are safe. Anything you copy on a Drobo array, as we call them, is protected because if one drive fails, you simply remove it and replace it with another one. I think these are terrific products. I'm so happy to have Drobo as a sponsor because I can tell you I use them all the time at home and here at work. And I think as a consequence of us talking about Drobos on TWiV, a number of other people have obtained them. I know Alan now is the proud owner.
Yes, I have a Drobo FS. Very happy with it. And one thing that I'm not sure if I mentioned, but one nice feature of it, when I was a little concerned, since it's it's a proprietary product, they've got their own system for maintaining the RAID array. And initially I thought, well, oh great, I'm going to have to use their control panel to interface with the device. But it turns out it's it appears on the network as a drive. And I've written a little bash script that synchronizes my files my way. And
nice.
So if you want to do that yourself, you can. If you don't understand what I just said, you don't have to worry about that either, because there's a very user-friendly graphical program that comes with it that you can interface with it that way. Or I understand you can set it up with Apple's Time Machine to do it that way.
You can do automatic backups as well.
Yeah.
They're very flexible. Really good product.
Can you partition it?
Yes.
Yeah, sure. Can partition it.
Yeah, what the Drobo does is it takes care of the RAID aspect of it. It manages these like a self-healing RAID array.
Wow.
But then it appears to the rest of the world, to your computer, or in the case of the Drobo FS, which is a network drive, it shows up on the network just like a drive. So I have two 2-terabyte drives in mine, which means it shows up as one 2-terabyte drive because it's duplicating.
Yep.
And as far as my computers are concerned, that is just a 2-terabyte drive. There's nothing unusual about it. So if you partition it, you do have to partition it with their tool, I think, to get it done correctly. But you can do it that way, or you can have it as one big block.
Got to get one, Mark.
Well, I've got a Western Digital, a Seagate, an Acom Data, and 2 Lessees on my desk, and it looks really dorky. Not only does it look dorky,
You could, if they're SATAs, you can put them all in a Drobo.
I gotta pop them out.
So if you go to bit.ly/TWiVDrobo2, there's a questionnaire you can fill out, and you'll be entered in a contest to get 50% off the manufacturer's suggested retail price on a Drobo FS, I believe. So check that out, Mark. Otherwise,
I'm absolutely going to match for you. I can use your promo codes.
Let's see, bit.ly/TWiVDrobo2 is the questionnaire, and the promo code is TWiVPod. That's a separate one. I just set up Dixon's Drobo, but he's gone, so I'll wait for him to come back to tell us about it. Anyway, we are really appreciative of Data Robotics for their support of TWiV. I believe this is our last episode with Data Robotics support, so we really appreciate what they've done, and hopefully they have further penetrated the scientific and medical world with their Drobos. Thanks, Drobo. Okay, let's do some email. I have a voice email. We get one about every 3 months by voice, so I'm going to play that one.
Hey, TWiV guys. This is Welkin Johnson. I'm an assistant professor in the Harvard Virology program up here in Boston, not too far away from New York. calling in because my favorite viruses and the viruses we study in my lab are the primate lentiviruses, and they're usually called the simian immunodeficiency viruses, or SIVs, probably inappropriately, but they're called it obviously because of their infamous sibling HIV-1. Anyway, I'm calling in because one of the interests in our lab is trying to understand how these viruses manage the feat of adapting to new hosts when they cross species lines. And so I was happy to hear on this week's podcast to hear Rich suggest that one thing that might be done is to look and see how the SIVs in their natural hosts, the African monkeys, how the viruses have adapted to cope with the MHC of those hosts. And I wanted to say this sounds like a great idea, and I'm pretty sure there are people who are looking into things like that. But the comment is also, and the reason I called, it's an opportunity to make an important point for many of the TWiV listeners, because I think it might, I think it deserves to be made. The point being that the naturally infected African primates are not analogous to human elite controllers. These animals are infected with virus, and sometimes they're robustly infected. And so the phenomenon in the natural host is not one of resistance or immunity to infection. And my guess actually is that MHC and antibody probably aren't the important players here. What's really so interesting about many of the African primates is that while they are infected, they don't appear to develop AIDS, at least not during their natural lifespans. And so they are almost what a classical geneticist might call classical geneticists might actually call the natural host of SIV a mutant that has genetically uncoupled infection from disease. At least that might be one way of thinking about it. So they are infected, and what's interesting is they've somehow gotten to this point where they tolerate the virus and don't seem to progress to AIDS the way humans who are readily infected with HIV-1 do. Anyway, that was my comment. I love the show, and I also just thought I'd mention that I am now hooked up to my new Drobo S, and when my graduate student walked into the office and saw it for the first time, she had the same reaction that Dick did. And I have to agree with her, it looks exactly like a toaster. Anyway, love the show, and look forward to hearing more podcasts. Bye.
Guys hear that?
Yes.
Great email.
Yeah, that's excellent.
So Welkin is going to be on TWiV in a couple of weeks. So he works not only on simian lentiviruses, but human endogenous retroviruses. So we'll get the word right from the expert's mouth.
Cool.
Second email's from Sharon. Hello. Are you still there, Marc? Ooh, we lost Marc.
Oh. No, sorry. My mic was off.
Okay.
Sipping on Coca-Cola.
Okay.
Sorry.
It's okay. Sharon writes, hello, fellow virus lovers. I first want to comment about Vincent's pick of the week a few weeks back, the book Polio by David Oshinsky. I am currently studying poliovirus in Julie Pfeiffer's lab, as you revealed many moons ago with a previous email I sent, and I received this book as a gift from my mother. David Oshinsky was in San Angelo, Texas, my hometown, and in the first sentence of the book, speaking about his book and receipt of the Pulitzer Prize and doing a signing. My mom attended the event and subsequently purchased a book for she and I. I love it. This is a wonderful historical perspective of the societal impact of poliovirus and impending vaccine arms race between Salk and Sabin that you don't necessarily get from PubMed. This book has been circulating in our lab for a while now, and I recommend it to anyone. I would like to recommend a pick of the week that I think hasn't been suggested yet. It's a PBS special called Naturally Obsessed. The film, about an hour long, is a documentary that follows 3 graduate students in the pursuit of their PhDs and the divergent routes they take. We watched the video for a lab meeting and all sympathized with the students in the video in regards to the rigor and disappointments of mountains of negative data of graduate school. It does have a happy ending. One more short mention. I have listened to every TWiV podcast and have heard you all ask several times if anyone is studying gastrointestinal, commensal, bacterial effects on viruses. No one to date has published on this subject, but keep an eye out. As always, I love the podcast. Thank you for broadening my virology knowledge. Actually, Naturally Obsessed, which will be my pick today, thanks to Sharon, was shot here at Columbia in the lab of Dr. Larry Shapiro. And the DVD just came out this month, so you can buy it. Put a link to it in the TWiV bookstore.
Negative data in graduate school?
Yeah, what's that about, right?
I never experienced anything like that.
No.
Never heard of that.
I joined a lab, and it took me 2 years to prove that the lab's model was wrong. And then, that was 2 years of negative data, and it saved the lab, I suppose, in a way of publishing that data point that we were striving to get. And then I sat down with my PI, and he looked at me and says, "Okay, what's next?"
Yeah.
He wasn't even frazzled. He had so many projects going on. And he says, "As long as you graduate before your hair is gray, then you're fine."
Well, also
my
learned.
entire thesis was kind of a negative result, too.
You had a wonderful positive result.
I demonstrated polio doesn't get into cells the way we thought it did.
I think that was a wonderful observation that stimulated the field for years.
Oh, good.
Yeah,
good.
no, I look at it. It depends how you view it.
Okay. I like to say that we knew less after my thesis than we did.
Well, that's the progression
science.
of knowledge. That's fine.
That's fine. Every question leads to more, right?
I have no problem with that. It's fine. The next email is from Andrew, who writes, "I'm a second-year undergraduate in genetics at Queen's University in Belfast, Northern Ireland. I've been listening to TWiV for about a year now, and I find it fascinating. I had not been much interested in viruses until I discovered your podcast and took an introductory virology module this year, but now I'm considering the field of virology as a long-term career. What caught my eye the most within my virology course is the idea of virus-mediated gene therapy, something that, and I could be wrong, I don't remember you talking about on the show. My sister is affected by type 2 spinal muscular atrophy, a condition which is caused by the loss of the SMN1 survival motor neuron 1 gene, and my question is, how far away are we from being able to successfully carry out gene therapy for genes such as this? My thoughts are that one of the main problems would be the size. From my basic knowledge, I know we have successfully put GFP into a measles virus and had it express GFP in ferrets, etc., but genes like the SMN1 gene are considerably bigger, 28 kilobase pairs as opposed to the 1 kb GFP. So what are the current and theoretical limits of gene size we can integrate into viral genomes? And finally, what would be stopping us from simply building our own viruses to deliver the genes? I've heard about poliovirus, which was built from scratch because the sequence was known, and it was simply ordered as primers and put together to create a live virus. But with lots of different strains of different viruses being sequenced and publicly available, wouldn't it be possible to engineer and build a fit-to-purpose virus? It seems logical to me that you could take a different sequence from wild viruses and create a viral genome of your own. Capsid protein from here, a reverse transcription enzyme from there, a replicase enzyme from somewhere else, and so on. Could it be possible that we could create a virus that would give a persistent infection of the appropriate tissues that shows little or no harmful cytopathic effect and delivers your missing gene? I would love to hear you talk about this on the show. Look forward to my TWiV fix every week, Andrew. Ooh, 2 good questions, Andrew.
Yes.
As far as I know, we are already doing gene therapy for a variety of disorders, immune deficiencies, neurological disorders, vision disorders. I was just in Florida talking with a fellow who is using adeno-associated virus to deliver a gene to people with a certain kind of vision disorder. They inject it right into the eye, and it can partially restore vision. So these mainly use lentivirus vectors, as we talked about in one of today's stories, as well as adeno-associated virus. There is a limit. There's an upper limit to the size of the gene that you can put in.
Did you say 28 kb?
Yeah, that's large.
That's
intron.
pretty big.
That's intron. Get rid of the intron. Just get the open reading
That
frame.
could very well be that includes intron. So, yeah 28 would be too much as far as I know.
It's not a peptide.
But then again, lentiviruses, you basically... you have to package it, and it's always the packaging limit that holds you back. So that's probably too big.
2 viruses.
Yeah. Now, as for making your own virus...
No, but you can make 2 viruses. Carry half the
Half
gene
in
and
one. But how would you get them together, though?
ligase.
No, the cell with ligase.
Homology.
Actually, there was a strategy, an AAV-based strategy years ago, similar to that, where they put two halves of a gene in two different particles, but never went
Often
anywhere.
proteins will reconstitute, right? There's a whole slew of protein interaction technologies, right, where you're studying how two proteins interact by making half of the transcriptional activator and then making the other half of the transcriptional activator. As it assembles or those proteins interact, it fuses the functional protein as a whole. Does that make sense?
Yeah, I understand.
Half the gene, the protein gets made as two half parts, but the protein comes together inside the cell, and then the two proteins come together and form the full protein. So you could, who knows, maybe you could do that. And that's pretty, 6 months to do, to figure out how to split them apart and make the peptides that will come together and form a complex.
Yeah, I understand. It's not a bad idea. But at the moment, we are limited. The upper limit, let's see. Well, in my textbook, I have a chapter on virus vectors. So let's see if I can quickly find the upper limits here.
As I said before, as a PhD student, I'd make 2 baculoviruses. And it co-infect, and the protein that was made was heterodimer, so 2 different peptides that would assemble.
Ooh, look at this.
Do that.
So adenovirus can go up to 38 kilobases. Adeno-associated, 9 kilobases. Epstein-Barr virus, 120 kilobases. Poliovirus, too small. Vaccinia virus, 75 to 100 kilobases. So you can get bigger incidents, but many of these viruses aren't being used as much. So mainly adeno-associated virus and lentiviruses. Lentis can take up to 18 kilobases, and adeno-associated viruses about 9 kilobases. So for the bigger ones, you have to use different viruses, but I haven't seen them.
I'm trying to find the mRNA length for SMN1, but I'm not coming up with it.
To see if it's shorter than the gene.
Yeah,
size,
everybody's got the gene length, and but
his second part, how could you make a brand new virus? See, I've always said you can't because you don't know exactly what to put together, but you could always try and hope that things change sufficiently so that they work together. So you suggested a modular approach, protein from here and from there. I do think that there's not enough compatibility depending on how far apart you take these proteins from. I think it's very hard to make your own virus. Nature has had years to do it, right?
Yeah, and I don't think that it's productive. I mean, I know all the hype now is around synthetic biology and it's going to solve everything. But we said that 10 years ago about sequencing genomes. So it's an interesting approach, but evolution's been working on this for a few billion years. And it's to the point where you've got this whole set of parts, and they're all organized hierarchically, even, and you can choose from all this pre-made stuff rather than reinventing the wheel. I think it's a much more productive approach.
Yeah, good point.
By the way, this is really small. Are we talking survival motor neuron 1?
Yeah.
It's really small. It's really, really small, and but it's got a lot of introns.
Yeah. Did you get an mRNA length on it?
No, I ended up in the database, the PDB database, protein database.
Oh yeah. It's like a 7 kilodalton protein or something, so it can't be a very
itty bitty little thing.
Oh, yeah,
so
that's easy.
That's perfect for this kind of thing. You just gotta choose the right splice variant.
You have to just get it to the right cells also. That's the thing.
True.
Yeah, and I gather SMN1 and SMN2 are made by the same locus, differentially spliced. But anyway, that should certainly be, in principle, it's doable.
Okay, Brent writes, "Hi guys, excellent podcast for an IT business analyst with no virus knowledge. Just wondering about flu/cold transmission in the household. Would sharing toothpaste be a valid way to transmit easily? I figure it would, i.e. person with a cold, toothpaste tube to brush, mouth to brush, next person has toothpaste tube to brush, brush to mouth, or is this paranoia? Does toothpaste kill it? Interesting. Would it be a high chance of catching the cold?" Well, I looked in my toothpaste tube when I got this. I didn't find anything that looked antiviral. It's mainly filler, really. And so I don't think it's any antiviral. But it really depends on how much virus gets on that toothpaste tube, right? And I suspect not very much. What do you think, guys?
A lot of toothpaste now contains triclosan, which is a lot of everything now contains triclosan because everybody wants antibacterial products and antimicrobials. And I'm just looking, I think that may be difficult for flu to survive. It certainly kills a lot of bacteria.
It's a phenolic product, right?
Yeah. It's in absolutely everything, including now, of course, all of our waterways. I'm personally not a big fan of it, but I wouldn't expect toothpaste to be a terribly hospitable environment for the virus.
Yeah.
Well, for standard sterile technique, right? You wouldn't be wanting to spread anything, like touch anything that's had contact with virus, right?
True.
So just to be safe, everybody in the family has their own toothpaste, right? A little small tube.
Yeah, it's an easy fix.
Yeah, it's very easy. Yeah.
Because the toothbrush did have virus. The toothpaste has virus. Now, did that virus survive or not?
Yeah.
That's how quickly, the other person, usually there's 3 people lined up brushing their teeth. So.
Yeah.
Right.
Well, we should do the experiment and get a grant to do it. Yeah, good idea.
Well, Crest might pay for it.
Yeah.
I got the virus. I got the toothpaste. It's easy to do.
Well, Crest might pay for it depending on your results.
Well, they have to put the money up front,
They might pay for certain results, right?
Yes.
That's not good. Our last email is from James, who writes, "Thank you for putting together a great podcast. My question refers to the podcast Gators Go Viral, in which you talk about viruses attacking cancer cells. Jaw dropper. I was wondering, and it might be a little early to talk about this, but is there any chance of evolution resistance in these cancer cells when you expose them to this virus treatment?" So in Gators Go Viral, we talked about using viruses to specifically kill cancer cells. Viral oncolytic therapy, it's called. And this is gaining momentum. It looks pretty good. So he's asking whether the cells could evolve to be resistant.
Yes.
Sure. Cells don't evolve as quickly as viruses, but you could imagine, you lose the receptor on the cell surface and they become resistant. I don't know how readily that would happen. It's a good question, and it's not something we'd know really until you start putting these therapies into a lot of people.
Yeah, and it would also depend on how quickly and reliably the virus kills to begin with, I think.
Yes.
Because if it kills 100% of the cells that it binds to, then they're not going to have a whole lot of chance. But if it kills 80% of them and the other 20% kind of struggle on for a while, then that might breed more resistance.
Mm-hmm. Yeah. So maybe we'll see in some of the clinical trials. These are all going in to
sometimes
bigger
you just
trials.
need to sorry to interrupt. We do have a little bit of lag here. Sometimes you just need to reduce the size of the tumor so it becomes resectable, right? So you cut it out, or sometimes just prevent the metastasis, so you prevent the spread. So not necessarily have to kill all the cells.
And the other thing is these are often done as combination treatments, right?
Right. So if you have liver cancer and the tumor is too large to resect, I guess you need to go with heavy chemotherapy, and you go in with go right into the liver, and you put a catheter in the liver. And this might be a simple way to reduce it from the outside and get it to a point where you can go in and cut it out. Right. That'd be amazing.
Yeah. Mm-hmm. Yeah. Well, James, I'm going to send your question to Grant McFadden, who was on that episode. We'll see what he has to say. But anyway, thanks for sending it. Let's do some picks of the week. Ooh. Mark, you got something in mind, or you want us to come back to you?
Sure. No, I got a pick.
All right, good.
So I don't know. Have you guys done the iPad as a pick?
We have not. You can be the first.
All right. Okay, so here's why I call this a science pick. One and very, very important aspect of science is writing. I mean, it's up to 50% of your time sometimes, or even 100% of your time during grant season is spent writing. And I tend to not like to sit at a desk too long. I've been waiting for years and years. I'm prone to developing deep vein thrombosis, which can happen from writing grants, which turns into pulmonary embolisms. So you want to stretch your legs every hour. But that being said, I've been looking for a tablet to write on, a really good mobile system where I can kick my feet up on a chair, so improve blood flow and stuff like that. Not that this is about blood flow and just walking around, but I've been looking for a really good mobile writing device. And the iPad, what's amazing about it, if you use their word processor Pages from Apple, the text rendering is fantastic. When you're writing on a computer, you're using a 3x4 screen that's horizontal in a landscape mode. It's not, and the screens, even a really good LCD, doesn't render text that well, and so it doesn't feel like you're reading a piece of paper. And so with the iPad, the resolution of the screen and the 4x3 format, but in a portrait mode, gives you this real tactile feedback that you get when you have a piece of paper in your hand and you're reading. Maybe it's not quite paper, but it's so much better than a computer screen that, I don't have to print out. So I write a page or 2 pages. I don't have to print it out and read through it on a piece of paper. It gives you that instant feedback of, here's what it looks like on the page, and the page layout looks really, really good. So I'm now doing all my grant writing on the iPad. That being said, which word processor do you use? Right? There's 3 main ones. Pages from Apple. There's Docs2Go, which is an old. They've been doing mobile word processing for ages, since the Palm Pilot. And then there's QuickOffice, right? And those are Word compatible word processors. Now, Docs2Go and QuickOffice both have really good file management systems, and you can access your Dropbox. I mean, Dropbox is the best way to get a file onto an iPad or onto an iPhone. So you can access your Dropbox, which is a must, by the way, in this process of writing on an iPad. You need a Dropbox. You need to install it. And then it can also access your Google Docs. So you can work and do word processing on the cloud directly with Docs2Go and QuickOffice. Pages can't do that. But Docs2Go, when you're moving up and down your document, it hesitates, and you move your finger up the screen, and it just blocks and makes you grit your teeth and say, I can't use this. This is just unusable. Go to QuickOffice, which is another competitor of Pages. It has a beautiful fluidity that you get from a mobile OS like Android or by iPhone OS or iOS now. But the text rendering it's like an iPhone app on an iPad. The resolution's about half, and the text rendering is just horrible. So use it for 5 minutes and just say, it can't be used. Pages, they've nailed it. The amount of word processing features, the text rendering, the feel of flipping through the pages, up and down through the pages of your document, and the feedback that you get while writing are absolutely fantastic. I will not write on my computer anymore. I'm writing all my grants on the iPad. A couple of things that you need, though, if you're going to set up do writing on an iPad, is you need your research, right? You need your PDF documents of your scientific literature. Now, I use something called Papers, which is an archive. It's basically iTunes for scientific papers. It has access to at least 8 or 9 different search engines, and it allows you to create folders where you drop all your PDFs, and then you can export EndNote or bookends references, basically. So that's where I archive my PDFs. But here's the problem. When you're in the library, you've VPN'd in, or you've proxied into your library, right? I proxy into the university that I'm affiliated with. It gives you access to all the scientific literature through all the scientific journals, right? But if you're in Safari on a mobile browser, you can't save them. There's no. It's like it's kind of, if you're on your iPhone or on your iPad, you can't save them. So there's an app called Goodreader. What Goodreader does is, when you're surfing using Safari and you get to your PDF loads, right? You've gone to the journal that you're interested in, glycobiology, whatever, and you got your PDF there. Then you go into the URL box and you put G in front of the HTTP. Then what it does is it instantly loads it up to Goodreader, and then you've captured the PDF off the internet. Papers won't allow you to do this because Papers can't handle a proxy yet. It's horrible proxy. I suspect that it'll get there, and when it gets there, you won't need Goodreader or Safari because you'll just do all your literature searches and papers. But he hasn't fixed the proxy yet. So the current workaround is use a combination of Safari and Goodreader. Both Safari's free on the iPad and the iPhone. Goodreader's like 99 cents, I think it is, and it's an ultimate file management system. It's really, really great. So if you combine Safari, Goodreader, papers, and pages, you have a brilliant writing environment. Leo Laporte, the anchor of TWiV, says that the iPad is simply a content consumption device, where I'll argue that in terms of reading and writing, it's a phenomenal content producer. It's fantastic for writing. So that's my pick of the week. It's Pages combined with Papers, Goodreader, and Safari, and you're a good, if you have a good proxy into university, you've got the ultimate writing experience, one that I think parallels going from WordPerfect 5.1 to Word, which was GUI driven. This is the next step.
So do you type right on the pad, or do you have an external keyboard?
It depends. I have an external keyboard, but I don't use it often. I write grants. I'm lying in bed, my head down on the pillow, looking upward, and.
So now, for the critical question for me, because this is my work environment for my writing, which I do for a living, will it run Emacs?
Emacs being what exactly?
It is the text editor.
Emacs.
I can't believe you're still using Emacs. I know a lot of people are, but
I was born in 1970. I don't know what Emacs are.
Oh, okay. I'm a text editor. I'm a text editor bigot. I would actually settle for Vim too, but that's going to be my sign to buy the iPad.
So you can. I've got a VPN that allows me to. What you do is you on your Mac. Before you leave work, you set the resolution of the screen to the same resolution as on your iPad. And then you can VPN in to your iMac and then control your iMac with your iPad. That way you can run Flash.
No, you could run it on there then.
So I could teleport into Emacs or Vim.
Yeah.
Okay,
sure. But then it's going to be a little bit sluggish. But as I said, I've watched Hulu TV shows on my iPad by VPNing into my work Mac. If that makes sense.
Yeah.
Okay.
But Alan, do you need a terminal to run Emacs?
No, it's it'll run in a window. It's window aware.
So you run it on a browser, or
I run it Vim or Emacs will run on just about anything.
Oh, so they're standalone applications.
Yeah.
Okay.
Yeah, you can download Emacs for the Mac, or you can download. There's a Mac Vim application that comes up in a window.
I'll bet someone's going to port it to the iPad.
I just did a quick search on this. Emacs seems to be a little tricky, but a lot of people appear to be finding ways to use Vim on the iPad. Yeah. So maybe I would switch.
I have an iPad, Mark. I love it, except I can't get my hands on it because the kids have it all the time. So I have not been able to try it seriously. But I think tonight I'm going to pick up Pages. I have Papers already. And I'm going to check it out because I would like to bring it with me when I travel and be able to write on it. That would be great.
Throw on Goodreader and you're set. Because Pages, Papers can't proxy yet, but Goodreader can, because Goodreader uses the system-wide proxy. Like you go into the settings, set up your proxy directly, and so Safari and Goodreader use that. Good
pick, and that's for science writing. There you go. I think that's great. Alan Dove, what have you got for us?
Well, I have a much milder pick of the week. This is just a frivolous little thing that I found amusing. It's a YouTube video. I guess the second week in a row I've done a YouTube video. I'll have to do something more.
It's okay.
More serious, a little heavier weight next time. But this is about a 30-second video called "The Bacterium and the Bacteriophage." It's a very high-resolution view of a bacteriophage infecting a bacterium, done in clay.
Claymation.
Yes.
No awareness. Oops. So, I've been playing it here, and it's
for your viewing pleasure after the show.
All right, excellent. Well, my pick is the Naturally Obsessed movie suggested by Sharon, which you've talked about. It was filmed here at Columbia in a lab right here, and I know some of the participants will put a link to it. You can get it on DVD. I think they will come by and show it to you if you want to show it to your graduate students or postdocs. I think they will give you a reduced rate to do that. So it's at naturallyobsessed.com. And that will do it for another TWiV. There are many ways you can listen to us. We're on iTunes, of course, the Zune Marketplace. You can get automatic updates of every episode if you subscribe at either of those two places. And, of course, if you go to iTunes, please leave a comment. It helps keep us on the featured page of the medicine section at iTunes, which is always good. You can also listen on the website, TWiV.tv. We have a download link. You can play it right there. Or if you have a mobile device, an iPhone, an iPod Touch, an iPad, BlackBerry Palm, or Google Android device, you can use Stitcher Radio to stream TWiV to your device. You can check that out at bit.ly/StitchTWiV. And you can always find show notes as well at TWiV.tv. If you like TWiV, tell everybody about it. We'd love others to listen in. TWiV is part of microbeworld.org, sciencepodcasters.org, and promednetwork.com, websites where you can find high-quality science content. And as always, send us your questions and comments to TWiV at twiv.tv. Marc, thanks for coming back. Great to have you, as always.
No problem. I really appreciate the opportunity.
I hope you like learning about viruses.
They're great.
Great stuff.
They're always a little scary, you know. That's that little edge.
Yeah, that's right. My student told me today that when she tells someone she works in a virus lab, she says there's one of two reactions. They either say, "Oh, that's cool," or "Am I going to catch anything from you?" So viruses have that effect. Marc can be found at twit.tv/FIB. That's the right place, right, Marc?
Mm-hmm.
Check
That's
out
right.
Futures in Biotech. Great science podcast. What's the next episode? Can you tell us?
It is a story of a man who was a homeless child in Italy and made his way to the Nobel Prize.
Wonderful.
It will be, he's on, his name is Capecchi, Dr. Mario Capecchi.
Oh,
He won the
yeah.
Nobel Prize with for his work on creating the genetic knockout of mice, which is an extremely important tool in modern medicine for genetics.
Yep.
And so he was homeless. He lived on the street for 4 to 5 years in Italy because his father had died. His mother was put into prison for passing around anti-fascist pamphlets. And he was surviving on toast crusts and coffee beans. And after 5 years, he was malnourished. He was in the hospital, and his mother found him. And his mother was American, and the family brought him to the U.S. And he went on, he had a fantastic education, did his PhD with James Watson at Harvard, and then went on to have a brilliant career in science. So he'll be on Friday, July 2nd at 11 a.m. Eastern Time, or 9 p.m. Mountain Time because he's in Utah, or 8 a.m. Pacific.
Wow.
July 2nd.
Sounds like a great story. Good.
It's an incredible story.
Yeah, we all know Dr. Capecchi's work very well. Yeah.
Yeah.
I had a professor in graduate school who was a successful guy, and he ended up being homeless. So there's the opposite path.
Wow.
Yeah, he died on the streets of New York of pneumonia. Can you believe it? This is a professor I had in graduate school. So not every story is the Cinderella story.
I had a professor who I saw carried out of the school by security because they thought she was a bag lady.
Oh,
and
my.
she was a phenomenal genetics teacher.
Wow,
She was phenomenal.
terrible.
Brilliant sense of humor. Very inward-looking person. You get her in front of a classroom of 150 people, she turned on with a charm that was incredible. But she didn't have the best hygiene.
Okay, that's
She's
terrible.
great.
Sorry about
She was
that.
fantastic. She was fantastic, though. I'll never forget her lectures. They were great.
Alan, thank you.
Oh, always a pleasure.
Alan is at dovedocs.com or alandove.com, where you can find short and clever sentences. I was just reading it this morning. I like the format of little things like, 1 or 2 sentences of cool stuff. I like that.
Well, it's kind of it's my own version of Twitter, Facebook, what have you, because I've tried those services and was dissatisfied with them for various reasons, and so I set up my blog to kind of do the same thing. So yeah, for people who are interested in my science writing and my professional life, you should definitely go to the DovDocs site and the DovDocs site alone. That's the one that's professional. And then my personal site, alandove.com, is just all the drivel that falls off my keyboard, plus everything from DovDocs. So if you want to read the whole stream, you can go to the Alan Dove site.
Alan, you might like daringfireball.com, all one word. It's a tech podcast. I'm sorry, it's a tech blog, but same idea. He puts, throughout the day, he posts a few sentences here and there with links to stuff that he likes.
Oh, cool.
And I want to emulate that format for my blog, but I haven't figured it out. He custom designed his blog software.
Oh, wow.
So that not every post gets a heading, not every post gets a title, which I like.
Well, I came up with my own WordPress theme for mine, but and it won't always be short sentences. I may post longer stuff on that as well.
Sure, mix it up. Yep.
Yeah.
And I'm Vincent Racaniello, and you can find me wherever fine viruses are sold. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.